Good morning, good afternoon, or good evening, depending on where you are in the world, and welcome to today's Bioeconomy and Tides webinar, Unlocking Scalable Oligonucleotide Manufacturing, Driving Commercial Success with Asymchem's Multi-Platform Solutions, sponsored by Asymchem and broadcast by Informa Connect. I'm Brianna Costafret, and I'll be your moderator today. We have just a few short announcements before we begin. This webinar is designed to be interactive. On the right-hand side of your screen, you'll find the Navigation Center. Here you can participate in the attendee chat and submit questions for our speaker. The slides will advance automatically throughout the event. If at any time you experience difficulties with the audio or advancing of the slides, simply press your F5 key or Command R to refresh your webinar console. If you experience technical issues, please type your issue into the Q&A area, and we will be glad to offer one-on-one assistance. Now on to the presentation. Joining us today is Dr. Wuming, Chief Scientist for Oligonucleotides at Asymchem. Welcome, Wuming. Please take it away.
Good morning and good afternoon, and thank you for joining today's webinar. My name is Wuming Yan, and I am the Chief Scientist for Oligonucleotide at Asymchem. Today, I will share how Asymchem unlocks scalable oligonucleotide manufacturing and how we partner with our clients to drive commercial success through our multi-platform solutions. To begin, let's look at the oligonucleotide landscape. Oligonucleotide therapeutics are growing rapidly, driven by the ability to precisely target disease at the RNA level. Importantly, the field is expanding beyond rare disease into large indications. At the same time, the modality landscape continues to broaden. Today, we see antisense oligonucleotides, a single guide aptamer, and an increased number of conjugated oligonucleotide designed for targeted delivery. As of the end of 2025, there are roughly 20 approved oligonucleotide drugs globally, with the market reaching about $7 billion and projected to exceed $7 billion by 2030. This rapid industry evolution highlights a key challenge. As programs move into larger indication, commercial success increasingly depends on scalable, reliable, and regulatory-ready manufacturing. As oligonucleotide programs move toward later-stage development and commercialization, manufacturing quickly becomes a key challenge. Those molecules involve complex chemistry and modifications, making scale-up significantly more difficult. While traditional solid-phase synthesis is well established, its scalability is limited as production volumes increase. At the commercial scale, maintaining robustness, impurity control, and reproducibility becomes more challenging. While raw materials supply, purification complexity, and the rising cost of goods add future pressure together, those challenges make it clear that scalable, well-designed manufacturing strategies are essential for commercial success. Here is an overview of Asymchem's oligonucleotide technology platforms. We support both traditional and non-traditional solid-phase synthesis, including fixed-bed, static bed, and fluidized bed solid-phase synthesis, allow us to address different scalability and process-efficient needs. Beyond solid-phase approach, we offer liquid-phase synthesis technologies, such as convergent liquid-phase synthesis and tag-assisted liquid-phase synthesis, which have overcome resin limitations, reduce PMI, and enable large-scale manufacturing. In parallel, we are advancing enzymatic and chemoenzymatic synthesis as the next-generation platform with long-term potential for sustainable ultra-large-scale oligonucleotide production. Together, those technologies form a flexible multi-platform foundation, support oligonucleotide development from early research to commercial supply. As of today, solid-phase synthesis remains the foundation of oligonucleotide manufacturing and an area where Asymchem has strong commercial experience. We support traditional fixed-bed surface synthesis, which rely on column-based synthesizer, and is well established for clinical and commercial production. To address scalability and mass transfer limitations, we also implemented step-bed and fluidized-bed surface synthesis, which move beyond synthesizer-dependent format and utilize reactor-based surface synthesis. Those approaches enable improved mixing, higher throughput, and more flexible scale-up while maintaining robust impurity control and GMP compliance. Together, those surface platforms provide a reliable, scalable foundation for commercial oligonucleotide manufacturing. As oligonucleotide programs move beyond the practical limits of surface synthesis, liquid phase and hybrid manufacturing approach become increasingly important. These platforms overcome resin and column size constraints, significant reduce solvent usage, and enable step change improvement in cost of goods, particularly for high dose and high prevalence indications. Within this framework, convergent synthesis improve effective, improves yield and impurity control for highly modified sequences, while membrane-assisted tag liquid phase synthesis enable efficient resin-free purification with lower PMI and faster cycle times. Together, those hybrid approaches provide the scalability, robustness, and sustainability required for large volume commercial oligonucleotides. This slide highlights Asymchem's another hybrid chemoenzymatic synthesis platform. In this approach, oligonucleotide building blocks can be generated through solid phase synthesis, liquid phase synthesis, or de novo method, and then assembled using highly efficient enzymatic ligation. By leveraging direct evolution enzymes and multiple ligation modes, include single-strand, template-assist, and double-strand ligation, we achieve high fidelity and strong catalytic performance. This strategy enables simplified purification, improved impurity profile, and flexible access to multi-ion products such as thiamine and single-guide RNA. Although chemoenzymatic synthesis offers a promising path toward more sustainable and scalable oligonucleotide manufacturing. Nowadays, conjugation has become a foundational technology for modern oligonucleotide therapeutics. At Asymchem, we support a broad range of GMP conjugation platforms, include GalNAc, PEG, peptide, and small molecule ligands, enable tissue targeting and improved pharmacokinetics. Our capability extends beyond conjugation chemistry to include customer linker design, tight control of conjugation stoichiometry, and robust purification strategy to remove unconjugated species. Combined with comprehensive analytical characterization, this integrated approach ensures conjugated oligonucleotides are manufacturable, scalable, and ready for late-stage development and commercial supply. As you can see, Asymchem is provide one-stop integrated GMP services across the full drug cycle, from drug substance and drug product development to non-clinical study, clinical supply, regulatory submission, and commercial manufacturing. We provide seamless end-to-end support with global quality system and decades of regulatory inspection experience. We help our partners advance program efficiently, compliantly, and with confidence. This slide highlights Asymchem fits appropriate CMC support across the full development life cycle, from preclinical studies through IND clinical phases and commercial supply. We support a broad range of oligonucleotide modalities, include ASOs, aptamers, siRNA, and diverse conjugates, with integrated capability in raw material, drug substance, and CMC strategy. Our flexible end-to-end solutions are designed to scale with your program as it advances toward commercialization. For the analytical service, Asymchem provides comprehensive analytical services across the full development life cycle, from method development and impurity analysis to validation, stability, and regulatory support. Our integrated analytics ensure product qualities, process control, and regulatory readiness from preclinical stage through commercializations. This slide just introduces Asymchem's strong regulatory experience. Initially, just from small molecules, so we gain extensive experience, then extended our experience and knowledge to the oligo part. So far, we have done about 50 IND submission for oligonucleotide and have one for later stage NDA submission. So this year we expect to have 2 to 3 more NDA filings for our clients. Regarding the agency inspection, we have a very good and strong record. I will not go to the single detail, but I would like to highlight, as of today, we have done more than 100 inspections successfully from agency and serve customer audits more than 300 per year successfully. Regarding the supply chain, Asymchem supply chain assurance across synthesis, purification, concentration, and lyophilization through qualified suppliers, dual source strategy, and tight quality control. We ensure consistent product quality, mitigate supply risk, and support reliable, cost-efficient commercial manufacturing. Also, Asymchem had capability and capacity to manufacture the phosphoramidite monomers. That depends on how complex the monomers and also the timeline of the clients. So if the starting material need long lead time from vendors, so Asymchem can involve to manufacture those monomers to short the timelines. Here shows Asymchem's proven expertise in solid-phase synthesis oligonucleotide across all development stage. Since 2018, we have supported more than 100 preclinical and over 90 IND programs with multiple assets advanced into later stage development and commercialization. Our commercial scale solid-phase synthesis platform offers approximately 1.5 metric ton of annual capacity and 14 GMP synthesis line and broad support for common chemical modifications. At Asymchem, our partnership model goes beyond that of a traditional CDMO. We engage earlier, start at the pre-IND stage to design scale-ready, platform-driven CMC strategy aligned with the molecule modality, dose, and long-term commercial needs. With integrated drug substance and drug product capability, dedicated cross-functional team, and transparent governance, we proactively manage risk and support our partners from development through commercialization. Our focus is long-term collaboration to enable speed, reliability, and sustainable commercial success. Here, just summarize the commercial impact Asymchem delivers to our partners. By designing scale-ready process and executing CMC activity in parallel, we help accelerate development timelines while reducing technical and supply risk. Our multi-platform manufacturing strategy supports competitive and sustainable cost of goods, reliable global supply, and strong regulatory confidence. Ultimately, this enables long-term value creation and durable commercial success for oligonucleotide programs. In summary, oligonucleotide therapeutics are rapidly expanding into high-volume, high-impact indications, making scalable and robust manufacturing a critical determinant of success. Manufacturing strategies must be designed with the end in mind, balance speed, quality, cost, and regulatory readiness from early development through commercialization. With Asymchem's multi-platform capability, global capacity, and partnership-driven model, we help reduce risk and enable long-term commercial supply. Let's partner early and build your oligonucleotide therapy from concept to sustainable commercial success. That's all I had for today. I would now be happy to answer any questions. I see that we already have a few questions coming in, and please feel free to continue submitting questions through the Q&A box on your screen. The first question is, what skill has Asymchem achieved for chemoenzymatic synthesis? For SciI, we have successfully completed GMP manufacturing at 100 gram scale. For other modalities such as single guide I and divalent SciI, we have achieved gram scale production to date. I hope that answers the question. Thank you. I see another question asking what's the typical timeline for chemoenzymatic synthesis approach. This is a great question. I'm pleasant to say that we have a very strong team and advanced tools that allow us to deliver targeted oligonucleotide products efficiently. Once the fragmentation strategy is finalized, it typically takes about 3 to 4 weeks, supported by AI-based prediction models. A demo batch can usually be completed within approximately 3 months from project initiation. Following that, GMP batch manufacturing includes full workflow execution, and the final release can be completed within about 26 weeks. That brings us to the end of today's Q&A section. If we were not able to address your questions during this time, I will be happy to follow up with you individually. Thank you again for attending today's webinar. Thank you.
And that's all the time we have. Thank you very much, Wuming, and we appreciate your time and expertise on today's topic. I'd also like to give a big thank you to our sponsor, Asymchem, as well as everyone in the audience. We appreciate your attention and participation. Within the next 24 hours, you'll receive a personalized follow-up email with details and a link to today's presentation on demand. Please feel free to invite your colleagues and peers who may not have been able to listen to the event. On behalf of Wuming and Asymchem, I'm Brianna Kosafret. Thanks for your time and have a great day.